welcome to the 2022 Microbiome Informatics webinar series. And this is a second year running. We did this last year as a sort of response to the pandemic. A lot of trainees seemed very hungry to get hands-on genome-resolved experience and understanding how to do microbiome informatics. And that was incredibly well received. Over 750 registrants from 22 countries around the world. And so we reached out again, focusing on early career researchers who are leading in different areas in this space to try to present hands-on 1.5 to 3 hour sessions talking through a lot of the different aspects involved in microbiome informatics. If you're here today, you know that there's no real textbook yet. Everything's being invented as we go, and it's a very lively and exciting space to be innovating in. I'm Matt Sullivan. I'm from Ohio State University, and I'm a professor in microbiology and civil environmental engineering. And I am involved in several leadership roles related to microbiome science at Ohio State. And I'm truly, truth be told, not a microbiome person. I study the phages or viruses that infect microbes. And so if sometimes my expertise is a little bit thin in some areas, please forgive me. So how do we get here, and how is it that I'm presenting to you about a microbiome informatics webinar series? I just told you I'm sort of the virus guy, so that seems a little bit odd, but I really was involved in microbiome science quite early. During my PhD with Penny Chisholm at MIT a long time ago, it was really the start of microbiome science before it was even called microbiome. This is back just studying microbes in complex communities. And this was arguably the epicenter of a lot of early environmental microbiology. At MIT, within a few doors of each other, was Martin Pultz, Ed DeLong, Roman Stocker, Eric Alm, and Penny Chisholm, all there together when I was training in my formidable years there as a very impressionable time as a PhD student and postdoc. And this was followed when I started my faculty position in 2008 at the University of Arizona, when Howard Ochman pushed me out of my comfort zone and encouraged me to teach a problems in genomics course, which his vision for and his suggestion to me for was to really push studying microbes in complex communities using genomics. And this was a time when we saw microbiome science, or the study of microbial communities, advance from gene markers like 16S, ribosomal RNA, to shotgun metagenomics, where you're studying the community using all the genetic repertoire available through deep sequencing. When I started my faculty position at Ohio State in 2015, I decided to teach a microbiome informatics course, which expanded really this metagenomic side. And I needed help because there's a lot to take on. And as I mentioned, I'm really the virus guy. And so Ben Bolduc and Sharif Dubdab have been teaching with me for a number of years, this microbiome informatics class. And this was during a time when metagenomics went from sort of gene-resolved science due to short reads not assembling very well, towards currently genome-resolved science, where you get lots of data allowing you to see large chunks of the microbial genome, if not complete genomes in many cases. And I'll just say that this has been an incredibly exciting set of transitions to see through the years, but it really is a community effort to teach in this space because it's such a fast-moving and exciting field. So that's sort of how we got here. I want to excite you a little bit about why even study microbiome science. And of course, since I'm a virus guy, I want to present some of the fun virus information, and then tell you a little bit about what is coming here in the coming weeks in this webinar series. And then come back a little bit to why Ohio State and why microbiome science is so exciting right now at Ohio State. So why microbiome? We hear about the human microbiome and its importance all the time. We got lots of exciting information coming out of studying the gut microbiota and cancer, lots of complex diseases that are getting different microbes ascribed to them as drivers and or influencers. There's also strain level variation of microbes that we're beginning to learn can modulate the efficacy of drugs and treatments that people get. And even in popular parlance, we're starting to realize there's an inner ecosystem each of us that's dominated by this human microbiome, more microbial cells than human cells in your body, 100 times more human microbial genes than human genes. So a really important genetic repertoire influencing our biology as humans. But beyond human health, microbes do a whole lot of other things, and just a very high level bring out some of that importance. Microbes in soils can modulate the biogeochemistry of soils and modulate nutrients that can get to plants. Microbes, of course, can be pathogens on plants, so they're important in agriculture in those kinds of settings. And if you've been breathing, and I hope all of you have, half of the oxygen that you've been breathing comes from microbes. Microbes in the oceans produce a lot of the oxygen on the planet and we're actually involved in the early oxygenation of the planet a long time ago when cyanobacteria invented photosynthesis. And as I mentioned, I do assume most of you are excited about microbes because that's why you're here. So I'm going to hint a little bit about what role do viruses play, because viruses arguably play big roles in all these microbiota. And so I'll focus on the oceans. And in the oceans, this would be a picture of seawater that's been concentrated and then stained with a DNA stain called cybergreen. And the small dots are viruses and the big dots are cells. And in the oceans, we learned that there's a lot of viruses in a drop of seawater. If you swallow a mouthful of seawater when you're out swimming, you've swallowed 50 million viruses. And don't worry, these don't infect you or even your human cells or your human microbiota. These are, of course, ocean microbes that are adapted to infecting ocean hosts under ocean conditions. And in the oceans, if we do tracer experiments, we see that about a third of cells are killed per day. So they play a large role in mortality in these microbes. Most of the viruses cannot be cultured, and that's because most of the microbes themselves cannot be cultured. And so cultivation is an excellent focused way to study these viruses, but it doesn't allow you to study community level impacts or ecosystem impacts very well. And if you do genetic transduction experiments in labeled systems, and this is just from a small set of numbers in the Florida estuary, and then extrapolate that to the global oceans, viruses move a lot of genes from one host cell to another, about 10 per day. A lot of the toxins we associate with microbial, like Bordetella toxin, Shiga toxin, cholera toxin, these are actually encoded by a virus in the genome of that microbe, a prophage. And those prophage-encoded toxins are really the drivers in toxic outbreaks. And a big challenge for many years has been, with all this abundance and potential roles of viruses, what are their impacts on ecosystems? And I'll just say this is going to be a kind of a one-slide overview of a whole lot of different studies. But the idea is that during my career, we've gone from counting dots here, getting that sense that viruses are abundant, knowing that viruses are about 1 to 10 times as many as the cells that are in seawater, to really getting a genomic map of them and using that genomic map to understanding them. So what does that mean? First, it meant 6 years of developing systematic quantitative sampling approaches. And so this is a lot of effort just to get highly quantitative data that can be used in broad ecological studies. And then once we have those systematic quantitative approaches, it turns out there was a sailboat, the Tara, which is going out as part of the Tara Oceans Consortium, to be able to go out and collect globally ocean viruses and all the way up to fish larvae. And in that expedition, we were able to catalog 200,000 DNA virus species and begin to map these using genome sequence space with machine learning analytics and gene sharing to be able to develop a genome-based taxonomy. And this is a big advance because most of what we saw, more than 90%, was brand new, didn't fall into any kind of categories in publicly available databases. When we explore those genomes, we studied and learned that viruses steal key host genes like photosynthesis genes, and these are termed auxiliary metabolic genes, or AMGs. And so virus-encoded auxiliary metabolic genes like photosynthesis genes can actually be a new way in which viruses are manipulating core metabolisms in the oceans. And then with these catalogs, we can layer the catalogs onto a global map. And this is the Tar Oceans transect, which started up in France. Each of those white dots is where the boat stopped for 24 to 48 hours. And then the end of the page axis is depth in the ocean. Blue water are areas of low carbon flux, and red and yellow are areas of high carbon flux. And so what we saw is that when we use a genes to ecosystems modeling approach, machine learning plus ecosystems modeling, and we ask the question, which organisms best predict ocean carbon flux? And you should care about ocean carbon flux because it's having climate change. The oceans absorb half of the carbon dioxide that we humans put into the atmosphere. So when we use this genes to ecosystems modeling, it turns out the best predictors are viruses, and by far, 89% of the carbon flux variation in the global oceans can be predicted by virus abundances. And so this is a very powerful technique, and it's suggested actually that a decades-old paradigm might not be the whole story, that viruses, when they blow up cells, may create aggregates which can sink out of the water column and cause a large carbon flux. And then the other part of this genes to ecosystems modeling is that we can actually pick out the most important viruses, the VIPs. This is variable importance and projection, but it's the same idea. These important viruses in the system. And so these VIPs mean we can figure out which handful of viruses should we do focused studies on. We can use those hypotheses developed from omics-enabled work in stabilized tope probing or other experimental methods to assess causation from these more correlative inferences. And at this point, there's a strong population-based science established for double-stranded DNA viruses, and it's all part of an ecosystem of apps and resources called iVirus, which is at ivirus.us if you haven't run across iVirus yet. So these are all DNA viruses. Recently, we've been looking at RNA viruses, and this is work from Ahmed Zayed and Guillermo Dominguez Huerta and James Wainaina, where they discovered that if we just survey the global ocean RNA viruses, we double the number of known RNA virus phyla. That's what's shown here in this tree, where the gray are known viruses and the orange/red are novel viruses. And when we use those then to map onto that ocean ecology, we can look at ecological drivers of RNA viruses as compared to DNA viruses. We can again find auxiliary metabolic genes, now new metabolisms that RNA viruses are manipulating. And we can figure out that RNA viruses infect important hosts and are also important for that prediction of global ocean carbon flux. So this is a broad omics-based toolkit, a subset of microbiome informatics that's virus-focused, and it really is the focus of my group and many others around the world. From those omics-based hypotheses, we now can develop model systems-based experiments to investigate questions further. And here, I'm just introducing in this slide the idea of virus cells, which are cells that have been infected by a virus. And this is Patrick Fortier's idea originally in the literature, where a virus infects a cell, creates a virus cell that's completely metabolically reprogrammed by the virus's genomic material. That leads to a change in resources in the cell that can impact the environment that it's around. So the ecosystem footprint of each cell changes when it becomes a virus cell. And you can measure these by measuring transcripts, proteins, and metabolites. And if you were to plot that metabolic state of the cell in this ordination plot, you can see that viral particles or virions look different than the host cells, uninfected host cells, and they look different than a virus cell. And this is relevant because if virus cells are totally different, and a third of the cells are infected at any given time, this means that virus cells could be really important for understanding ecosystems. And so the folks listed on the left here have really driven some exciting work in this space, looking particularly using the Tar Oceans data to figure out which of the viruses are most interesting to study, and then using a time-resolved multi-omics toolkit to study how phage specificity and environment change across cells, how infection efficiency changes across cells, and how, in this case, cyanobacteria change under phage infection with and without a protist predator around. So that's why microbiome science, with a strong virus influence due to my background. I want to next shift to the webinar series and what's coming. And this is an overview. So this is meant to be Tuesdays from between 11 a.m. to 2 p.m. Eastern daily time. And that window may or may not be used, the whole of that window used each time. And you can see the dates listed there. We tried to basically start now and go every week to August 2nd, and then take a break for the month of August and start up again in September. Today, you're hearing about this introduction to microbiome science and to Ohio State and our role in microbiome science here from me. And next 2 weeks, you'll be hearing from Sharif Dubdab on high performance computing, an introduction to CHIME and mother and ecological statistics. After that, you'll hear about advanced ecological statistics from Ahmed Zayed. And then Dylan Cronin will start with some of the initial genome resolved metagenomics processing, where you work from reads through various binding strategies to get to high quality metagenome assembled genomes, or MAGs. And then we'll have a 1 month break for the rest of August. We realize a lot of people are on holiday at that time, so we'll come back early in the fall semester, at least those of us in the Northern Hemisphere, with DRAM, which is a scalable way to functionally annotate and pathway contextualize information from metagenome assembled genomes. So this is really genome resolved inference that'll be led by Caleb Orton and Rory Flynn from Kelly Wrighton's group in Colorado State. And then Christian Rinka will present on the GTTB, which is using genomes to do taxonomy and phylogenomics. And then we'll hear from Ben Bolduc on the basis of what's at iVirus and its adaptability, these apps and ecosystem resources that are either on the knowledge base or KBase or on Cyverse. So that's the whole of the webinar series. And again, it'll be on, to keep a regular time, on Tuesdays from 11 a.m. to 2 p.m. Eastern Daily, except for Christian is currently in Australia, and so his will be a slightly off time, which will be clearly delineated through the webinar process. I want to take a step back and recognize this is really an effort that we've tried to make complementary to other microbiome training opportunities we know that are out there. And so I want to share some of these. In case you don't know, the Cyverse and KBase, Anvio, the JGI, Chime2, these are all platforms and toolkits that are relevant for microbiome science, and they all themselves try to hold regular training opportunities. And so if you're interested in the Microbiome Informatics webinar series here, and you want to get more in-depth training or look at using other platforms, this would be an opportunity to look into training with some of these other microbiome platforms out there. I also want to give a kudos to the Microbiome Centers Consortium, which has been running since March 11, 2021, sort of an early career researcher microbiome seminar series. That's on Thursdays at 2 p.m. Eastern Standard. And that's been running for quite a while now. And we were in fact inspired by that in the virus community and have launched a similar one. Ours is not every Thursday. Ours is once monthly on the second Wednesday. And that's at 10 a.m. Eastern Standard. And that's presented by a series of virus-related groups. There's also the Virus Ecogenomics and Applications, or VEGA, the DOE Joint Genome Institute is running. They hold great viral ecogenomic focused community opportunities for workshops and symposia. And keep an eye out for the Microbiome Centers Consortium has an education committee. We've been centralizing educational resources to try to create a central portal for people to be able to come to, researchers to come to and learn about different training opportunities and resources that are out there. If you know of any others, please also let us know. We'd love to hear about other sort of complementary microbiome efforts that we have overlooked or not seen or not heard about yet. We'd like to create a real community spirit and atmosphere across the microbiome sciences. So we decided at Ohio State to launch this, and what's been fun for me is to realize that there's really been a long history of microbiome science at Ohio State, and we have an exceptionally interdisciplinary research support program on campus. So I'm going to wrap up in the last 15 minutes or so sharing some of that with you. And I'm going to start from this year, 1997, when I think of, that was for me the moment that I thought, wow, microbes are incredibly complex. People were just using ribosomal RNA sequences to start to explore the microbial dark matter or the microbial universe that's out there. And this sort of 3-domain tree was really pretty solidified after a couple of decades of being around. And that year is also the year that one of the first genome sequences for a microbe was sequenced, and it was done at Ohio State by John Reeve and Chuck Daniels and others. And this particular microbe was sequenced because of its importance in making methane from carbon dioxide, so important in a bioenergy set. As well, there is an RNA center at Ohio State that was led by someone who probably started the RNA world hypothesis with some real substance. That's Tina Hankin, when she published in the early 90s the idea of riboswitches, the idea that RNA could have a role besides just encoding as mRNA between DNA and proteins, and that RNA can really have a regulatory role. And that's led to a large expansion in thinking about early RNA world hypotheses, as well as other functions, regulatory functions for RNA. And then Juan Alfonso is the director currently of the Center for RNA Biology at Ohio State, and he actually had Norm Case on his PhD committee, so he had a connection to microbial communities a long time ago, and is really thinking at a creative interface between RNA biology and community context in a lot of his work looking at modifications. And then Ohio State had, as far as I can tell, one of the earliest microbial communities classes out there. Both DOE and NSF funded a microbial and physiology and diversity course from 1995 to 2000. And this was really pretty early in the game. And for these reasons, I think because of this long history, Ohio State has a real breadth of environments that we study. So a lot of times when we talk about microbiome science, we often think about the human microbiome. And I think a lot of centers are very focused on the human microbiome. And not only at Ohio State are we focused on the human microbiome, but also other mammals, other mammalian microbiota, but a lot of other environments, from soils in the oceans to the air that you might breathe around you in indoor environments, cockroach microbiomes as a way to study actually for human health, the evolution of microbiomes, even microbiomes on the International Space Station, and other extreme environments like hydrothermal vents or ancient ice cores, which have virtually no biomass, or microbiomes in communities or coral reefs. So lots and lots of environments, and that's the hallmark of Ohio State's microbiome community. And this has led to some exciting interdisciplinary work across 3 different entities: the Infectious Diseases Institute, the Center for Microbiome Science, and the new Emerge Biology Integration Institute, funded by NSF. And so the Infectious Disease Institute is part of our microbiome research landscape at Ohio State, and it has many different thematic programs, but one of them here highlighted in the red circle is microbial communities. And there's a large and vibrant group of faculty that are part of the microbial communities thematic program in the Infectious Disease Institute. And I think an exemplary way that the IDI has just flourished is in its interdisciplinary response to COVID. And it started when we stood up an applied microbiology services laboratory. We stood up COVID testing, and this saved Ohio State probably $100 million, because there's many, many students, faculty, and staff that needed testing for the last couple of years, and by doing that in-house, that was a huge opportunity. Now, those data could also be plugged in and complemented by monitoring at the level of wastewater and environmental reservoirs. And in fact, this group, the eScout group, surveyed many kinds of animals to look for reservoirs, and many of them were blanks. But you may have seen in Nature right before Christmas last year was a publication that they put out on white-tailed deer being a large reservoir for many different variants of COVID. And so that has changed the way that we all think about COVID epidemiologically. There's also practical aspects in this response. It turns out police cars, if someone were sick, how do you disinfect them between people who are detained? And so there was some press in the New York Times on the solution for that, where we'd figured out that you could just heat up the car with the existing kind of capabilities that the car had to be able to disinfect the car. Indoor surveillance is also pretty sophisticated here, particularly due to the work of people in indoor microbiome. And it turns out of all the kinds of surfaces in your indoor space, floor dust is one that collects an awful lot of COVID. And so, it's, I guess, keep things clean, be sure to dust. And then in a comprehensive viewpoint for disease context, there was an NIH-funded Center to Stop COVID that has been led out of the medical school. And when you put all these together, the IDI has been running an epidemiological modeling group for a couple of years now, since the initial outset. And this has allowed Ohio State to make scientific, data-informed decisions about best practices on campus. And this is a really powerful example of the complexity of a problem that you can handle with an interdisciplinary microbiome science solution. The second entity I'd like to highlight is the Center of Microbiome Science. And this was launched in February 2020. And a month later, everything was kind of shut down due to COVID. So it was an interesting time to launch, but the vision is to be able to really empower microbiome science for the design and prediction of microbial communities in animal, plant, human, environmental, and engineered systems. And we do that through what we call the 4 C's of compute, curriculum, capabilities, and community. And this is a very vibrant and robust community of 109 faculty across 9 colleges at Ohio State. And in just since February 2020, we've already, through compute, built out quite a bit of supportive infrastructure with greater than 100 apps and resources on the state of Ohio's high-performance computing system called the Ohio Supercomputer. We've also built out a 7-course training track for curriculum that no student would take all 7 courses. It's built with a few core courses and several courses to allow people to choose their own adventure through microbiome science, depending on their starting point and their final interests or goals. And to support researchers at Ohio State that are doing microbiome science, we have training-led working groups where weekly there's a trainee available or multiple trainees available to answer questions as people are troubleshooting challenging elements of microbiome science. For capabilities, we've built out a microbiome platform. We've got very cost-effective ways to generate data, and we also complement that with a core set of analyses, so data generation and analyses combined, to allow researchers to come in and not be lost in the pains of trying to get through data, but instead be handed data that really allows them to be the biologist and to interpret and make the most of those microbiome science capacities. And this is not a 16S-based platform. This is all genome-resolved metagenomics, metatranscriptomics, et cetera. We're not doing any 16S work at Ohio State. We're farming that out to the many opportunities that are available. This is focused on building out genome-resolved microbiome science. And then as a secondary capability, we're developing novel, innovative approaches for high-throughput phage capture and characterization as a way to modulate the microbiome to get at that design piece of our mission. And for community, we've got training workshops that we do, which are numerous and involved in multiple areas. A seminar series for the microbial communities community on campus, the microbiome community on campus. We do monthly mailers, which often highlight new scientists who have come to campus to help them get known on a big campus like Ohio State. And we do faculty mixers and trainee mixers that allow the group to meet and socialize, particularly now we're coming out of COVID. And we also try to connect with other groups regionally and globally through organizing meetings and other efforts. So beyond the Infectious Disease Institute and the Center of Microbiome Science, we've recently gotten an NSF-funded Biology Integration Institute called eMERGE, which is to study emergent ecosystem response to change, and is really focused on this particular thawing permafrost site in northern Sweden, which is a long-term ecological research site called Abisko. And at that site, there's lots of instrumentation and decades of study, and it's an actively thawing site. And so our interest here is to study with the full toolkit of different ways to study those soils, what the microbiome impact is on climate change gases and vice versa. How is the microbiome waking up as those permafrosts are thawing? I think this is really exciting because it's also, I would say, at the forefront of pushing microbiome informatics, pushing microbiome science through creating a framework for biological integration, in particular, what we call the genes to ecosystems framework, which really starts at the molecular biology level of sequencing DNA, RNA, and or measuring proteins. So metagenomics, metatranscriptomics, metaproteomics, and trying to connect those all as a readout of cellular processes and cellular functioning. Who is there? What are they doing? And then working on integrating that into a population context, a community context to look at interactions, metabolites that are passed between organisms, viruses are part of that equation. And then in the community context to the ecosystem context to really get at system outputs. And this was, in truth, sort of a decade of work prior to the eMERGE Biology Integration Institute that was called isogeny for isotopes and genes. And isogeny was a DOE-funded systems biology program for about a decade that studied this part of the loop, genes to ecosystems. And now with eMERGE, we're trying to close the loop to go from genes to ecosystems back to ecosystems to genes. So we're interested now in also understanding what we call the 3 A's of adaptation, assembly, and acclimation to understand the feedbacks of those ecosystem fluxes, ecosystem changes, and how they change the underlying microbiota in those interactions. This will lead to an understanding not just of what ecosystem outputs are, but how they're changing. And that is important because it can lead to a predictive modeling capacity, and there's multi-scale modeling involved in this, including trait-based modeling and larger regional scale models. This is, again, all focused on a single site and a single problem here of climate change microbiology. So just to wrap up, I've tried to emphasize for you, why microbiome science, and I've also flagged my imposter syndrome, I guess, that I'm really the virus guy, so I feel a little funny standing up here and trying to talk about microbiome science, but I have taught in this space for over 10 years and been thinking about microbiome problems and working alongside great microbiome scientists for a long time. If you actually like some of the viromic stuff, we have several openings. I would love to try to see if there's anyone who's interested in joining the team. We gave you an overview of the webinar series that's coming up here. That'll be going every Tuesday until August 2nd, and then we'll take a month break for the rest of the month of August, and come back in September to finish with the rest of the seminar series. And I've tried to give you a sense for why Ohio State, where we sort of launched around the same time as this exciting 3-domain tree of life that Norm Pace and Carl Woese put forth, and that changed everything in microbial diversity, microbial ecology. And at Ohio State, that we've had a long history of studying microbial communities and microbiome science, and this has led to some really nice interdisciplinary entities, the Infectious Disease Institute, the Center of Microbiome Science, and the eMERGE Biology Integration Institute. And I just want to close by saying thank you to Ohio State and the NSF who are supporting the faculty and staff that are putting together these webinars to try to create a nice training environment for everyone to be able to develop their cutting edge toolkit in microbiome informatics and microbiome science. Thank you.